Okay, this is chapter 13, part 3, and we have talked about sexual life cycles in part 1. We talked about what happens in meiosis in part 2, and now we're going to talk about some of the ways that we get variation, genetic variation through meiosis. Genetic variation is really important because that's what allows us to all have little differences. If we're all identical, there could be anything come along and just wipe us out very easily, and our species would go extinct. We wouldn't have people anymore. But because we are genetically diverse, we're able to use different food sources. We're able to survive diseases. We have a stronger ability for our species as a whole to survive. So the first thing we're going to talk about is crossing over. So during interphase, your sister chromatids are held together by cohesins along their whole length, and non-sister chromatids can be broken at precisely corresponding positions. So here you have your dad's DNA, here you have your mom's DNA. They're both able to break at this spot, swap places, and end up with a chromosome with dad's DNA with a little piece of mom's chromosome with a little piece of dad's at the end. You have this zipper-like structure called a synaptonemal complex. Okay, that's a big word, synaptonemal complex. Your synaptonemal complex holds your two sister chromatids together. It holds your homologous pairs together. So your sister chromatids are held together by cohesins, these sister chromatids held together by cohesins, and these homologous chromosomes held together by a synaptonemal complex. That's just a little zipper that holds them together. During synapsis, you break DNA from one piece and attach it to the other. So both homologous chromosomes are going to break and swap a piece of DNA from mom to dad or from dad to mom. So you end up with a chromosome that is a unique combination of genes instead of just straight from mom or straight from dad. Let's compare mitosis and meiosis a little bit. So mitosis conserves the number of chromosome sets, and meiosis reduces the number of chromosome sets. So in mitosis, you go from haploid to haploid. In mitosis, you go from diploid to diploid, or you could go haploid to haploid. In meiosis, you go from diploid to haploid only. So in mitosis, you produce cells that are genetically identical to the parent cell, and you're going to have two daughter cells. In meiosis, you produce cells that are genetically distinct from each other and from the parent cell, and you end up with 4 copies of each gene. So here you have a cell starting mitosis, and at the end you have 2 identical daughter cells. Here you have a cell starting meiosis. At the end you have 4 daughter cells, and they are each a unique combination of genes from that original cell, and they are each distinct from each other, and they are distinct from that original cell. 3 events are unique in meiosis, and they are all going to happen in meiosis 1. So first is synapsis and crossing over. Synapsis and crossing over occurs in prophase 1. That is important to know that it occurs in prophase 1. In synapsis and crossing over, you have homologous chromosomes that physically connect and exchange genetic information with each other. Also in meiosis 1, we have homologous pairs at the metaphase plate. Instead of just, each gene or each chromosome lines up with its sister chromatid, here you have homologous chromosomes also. So not just sister chromatids, but homologous chromosomes. And then separation of homologs during anaphase 1. So instead of separating into individual genes, you are separating into homologous chromosomes. You are separating the homologous chromosomes and keeping together your sister chromatids. So mitosis occurs during interphase. Oh my goodness, I am just losing all my words today, aren't I? So in mitosis and meiosis, DNA replication occurs before interphase. Back up and just start this page again. Okay. DNA replication occurs during interphase before mitosis in mitosis, and occurs during interphase before meiosis 1 in meiosis. In mitosis, you have 1 cellular division, and that includes prophase, prometaphase, metaphase, anaphase, and telophase. In meiosis, you have 2 divisions. You have prophase 1, metaphase 1, anaphase 1, telophase 1. then prophase 2, metaphase 2, anaphase 2, telophase 2. Homologous chromosomes are not put together in meiosis. In mitosis, they don't have to line up. But in meiosis, you have homologous chromosomes that meet together during prophase 1, that little zipper synaptonemal complex that holds your homologous chromosomes together during prophase 1. You also have crossing over between non-sister chromatids, and that'll give you chiasmata, and your pairs are held together due to sister chromatid cohesion. In mitosis, you end up with 2 daughter cells, and they're each genetically identical to the parent cell, same number of chromosomes. In meiosis, you have 4 daughter cells. They are always going to be haploid and genetically different from the parent cell and from each other. Mitosis allows an animal or plant, a multicellular organism, to arise from a single fertilized cell. Mitosis allows cells to produce more cells for growth and for repair. In some species, asexual reproduction occurs by mitosis, and it also produces gametes in the gametophyte plant, so only when we're talking about gametophyte plants. In meiosis, this produces gametes in animals. It produces spores in sporophyte plants. So if you remember when we talked about alternation of generations, spores are produced by the sporophyte plant. Meiosis reduces the number of chromosome sets by half and introduces genetic variability among the gametes or spores. Okay, some different ways we have genetic variation, and genetic variation is kind of what allowed sexual life cycles and what contributes to evolution of our species and into even evolving into different species. So mutations or random changes in an organism's DNA are the original source of genetic diversity. So sunlight shines on DNA, it can mutate it. Different carcinogens and mutagens can mutate your DNA. Sometimes DNA just doesn't copy perfectly and it mutates. Mutations will create different versions of genes called alleles. So the gene for blue eyes, the gene for green eyes, the gene for brown eyes, the gene for all your different eye colors are different alleles. They're all eye color genes. They're all the same type of genes, but they're different versions. They're different eye colors that are different pigments that are produced. And reshuffling alleles during sexual reproduction produces genetic variation. So the behavior of chromosomes during meiosis and fertilization is what allows for most of that variation to arise. And so we have 3 things that happen during meiosis that can lead to variation. One is independent assortment of chromosomes. And that's what we see happening in this picture. So you could have all of dad's genes line up on one side and all of mom's genes line up on one side, which means after meiosis 1 that you have cells with all dads or all moms. You could also randomly have them shuffle with dad's side here and mom's going to here, or mom's chromosome 1 and dad's chromosome 2 over here. So that's an independent assortment. Dad's genes aren't forced to go this way and mom's genes aren't forced to go this way. It's just chromosome 1, dad's or mom's goes to either one. Chromosome 2, dad's or mom's goes to either side. Crossing over, we've already talked about crossing over when ends of chromosomes can, swap, break off, and reattach to the other chromosome. And random fertilization. So at the end, you have all these different combinations of eggs. They're not all going to go on to produce, you are not going to have 8,000 or however many children. You're probably going to have a couple. And it is a random decision of which egg gets fertilized. It's a random decision of which sperm fertilizes that egg. Okay, so independent assortment of chromosomes. An independent assortment, I kind of just summarized that, but let's read this anyway. Each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of other pairs. The number of combinations possible when chromosomes assort independently is this 2 to the n, where n is a haploid number. So because we have 23 chromosomes, 2 to the 23 means there's 8 million combinations of chromosomes. So if that was all that happened, you could make 8 million different genetically distinct eggs, or 8 million genetically distinct sperm. Crossing over produces recombinant chromosomes. So that is this chiasmata, there's a word we've used. The chiasmata is the point of where they are crossing over. And you have this recombinant chromosome. That means the DNA came from two different sources. So you have some of the DNA from dad, some of the DNA from mom that were recombined. They broke and combined again. They're recombinant. So they combine DNA from each parent. Crossing over contributes to genetic variation because now instead of having just dad's chromosomes or mom's chromosomes, you can have just dad, you could have just mom, or you could have a chromosome that is a mix of both. And it can be a little mix or a lot mix. In humans, an average of 1 to 3 crossover events occurs per chromosome. So times all the chromosomes you can have a lot of crossing over, giving you much more variation. Let's watch a little video about this.
In sexually reproducing organisms, 3 processes lead to most genetic variation: independent orientation of chromosomes in meiosis, crossing over of chromosomes in meiosis, and random fertilization. Each pair of homologous chromosomes consists of 1 chromosome inherited from the father and 1 from the mother. Here we have color-coded them blue and red. Each pair of chromosomes lines up independently of the other pairs in metaphase 1 of meiosis. Here you see one of the possible arrangements and outcomes. There are 2 different ways that each chromosome pair can line up. That means that in the organism shown here, with the diploid number of 4, independent orientation of chromosomes at metaphase 1 can produce gametes with 4 different combinations of maternal and paternal chromosomes. In a human being with 46 chromosomes, more than 8 million combinations are possible. Now let's look at how crossing over creates even more genetic variability. During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of 2 non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from 2 parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes. Independent orientation and crossing over occur simultaneously during meiosis, multiplying the number of genetic variations among gametes. Because each pair of chromosomes lines up independently, and crossovers can occur almost anywhere along each pair of chromosomes, it is possible for a human being to produce an almost infinite variety of gametes. A sperm fertilizes an egg, producing a zygote. The random nature of fertilization adds to the variation arising from meiosis. Each parent is capable of producing a huge variety of genetically different gametes. The number of possible combinations among their offspring is staggering. Theoretically, one human couple is capable of conceiving a number of genetically different offspring that is far greater than the number of humans who have ever lived.
Okay, so those different ways we talked about genetic variation. I do want you to be able to tell me, if I ask you, what are ways that meiosis contributes to genetic variation? You need to be able to list those for me, just as a list, crossing over, independent assortment, random fertilization. But you also need to explain to me what each one of those means. So if I ask you, what does it mean when, parent chromosomes split and recombine with each other, you need to be able to tell me what it is. Or if I ask you, what does random fertilization mean? You need to be able to describe those for me. So know that in general what they are, but also be able to describe them a little bit better too. Okay, random fertilization adds to genetic variation because any sperm can fuse with any ovum with any unfertilized egg. So the fusion of 2 gametes, each of which have 8.4 million possible chromosome combinations, produces a zygote with about 70 trillion diploid combinations. So any man and any woman could produce 70 trillion genetically distinct children, which is far more than you're ever going to have. I don't think you could make it even to 70. So crossing over adds more variation, and each zygote is going to have a unique genetic identity. Natural selection will result in accumulation of genetic variants that are favored by the environment. Sexual reproduction contributes to genetic variation in a population, which originates from mutations. So first you have mutations, make different alleles. Then you have these three different ways that meiosis can give you unique combinations of alleles. And then natural selection will allow those alleles that are best favored to the environment to flourish. So we end up with individuals that are more adapted to their environment than they would have been without any of these mutations. There are animals that reproduce asexually, although they are quite rare. So things like the diploid rotifer, this is an animal that reproduces asexually. And these organisms have their own way of developing genetic diversity. So these asexually developing organisms can incorporate foreign DNA from the environment. So they pick up foreign DNA, and that foreign DNA allows them to be genetically diverse. And if that foreign DNA helps, the natural selection will tend towards that. And if it's neutral, you'll just get more variations built up. And if it's harmful, then that foreign DNA won't be carried through. We'll have more genetically diverse organisms that are able to survive in more diverse circumstances. Okay, so just to end, let's talk about meiosis in an animal cell. Prophase 1, you should be able to tell me what goes on in each of these stages. Prophase 1, you have sister chromatids lining up with their homologous chromosomes. You're starting to form spindle microtubules. It's during prophase 1 that you can develop these chiasmata and have crossing over. At metaphase 1, sister chromatids with their homologous chromosomes are lined up on the metaphase plate. At anaphase 1, sister chromatids are still attached, but separate to homologous chromosomes separate to opposite ends. At telophase 1 in cytokinesis, you reform your nucleus, you form two cells, either with a cleavage furrow or a cell plate. And then without copying DNA, you go straight into prophase 2, when you dissolve that nuclear membrane and start to have your centrosomes developing microtubules to attach back to your chromosomes. Metaphase 2, you have sister chromatids lined up at the metaphase plate. Anaphase 2, sister chromatids separate, so now you have one copy of each gene. And then in telophase 2, in cytokinesis, you develop your nuclear membrane, and you end up with 4 daughter cells. Each of your daughter cells are haploid and genetically distinct from each other. Okay, so that is chapter 13. So at this point, you should be able to discuss mitosis in pretty good detail to me. You should be able to discuss meiosis in pretty good detail to me. You should be able to compare and contrast mitosis and meiosis. And you should be able to describe to me how meiosis causes genetic diversity and why that's important, and describe different sexual life cycles. If you have any questions, as always, you can contact me. And I'll see you later. Bye.